The Wi-Fi cut out 10 minutes ago. That's it. The guest Wi-Fi cut out. Thank you for saving me. I appreciate it. All the rest went very quickly. But I did check the YouTube video is understandable. So that's really interesting. So if you feel like you've missed any gems of wisdom, they're all recorded. And I'll attach the link to the workshop page so you can revisit it and soothe yourself to save at night.
This room already has that. Well,
This room?
I took my pre-qualification exam on this room.
Oh, sorry. You're sure it's this room and not the next?
No, I'm sure it's this one.
The furrows in my nails made.
Been here for a really long time. Almost 8 hours.
You still got your PhD here?
No, that was a great pre-qualification. So you can get out from the first year.
So, were you DVM?
No, I'm in pharma.
Okay. All right. Let's see. Make life. So go to your computer view. Put your number. You already So now, now all the things that do see, everything should work. Okay. And not everything is working super. All right. So let's talk about full versus relative paths. So the CD command, this change directory command, takes an argument that's either what's called a full path or a relative path. Either one is a valid directory name. A full path, there's an analogy a little bit down below. A full path is like a GPS coordinate. Where you are on the computer, the full path unambiguously identifies a place on the computer. The problem with them is they can be quite long, because they go all the way to the root of the file system, and then they specify your user and your directory and this and that. So most people use shortcuts. So if you type, just to show you the difference between these, if you type, if you type CD and you get your help directory, which would be something like users T. If I type CD data and I do an ls select, I'm in this directory, users T data. If I do CD and go back to my home directory, and now I type CD/user/T/data, That's what's called a full path because it starts with a slash. And that puts me in the same place I was before, but I have to type more to get there. So the relative path says go here relative to where you're standing right now. And the absolute path or the full path says go to this location independent of where you're standing. And so when you're doing data analysis, it's typically nice to arrange things so that you say, well, I'm in my data directory, and my analysis directory would be something like /results. And then results will have a full path, but you never need to worry about where it is. You just know relative to your data directory, it's all one directory up, and then directory results. There's actually a lot of benefits that I can't fully convince you of right now, but imagine that you're working on one project, an RNA-seq project, and you put all your data in data and all your results in results, and you write scripts. You automate the analysis of the data to go to results. If you put that under a directory project 1, then later on you acquire a new project, because as you'll quickly find out, if those of you that are already doing sequencing data analysis, as soon as you know how to do anything in the UNIX shell or anything bioinformatics, you're the lab expert. So you'll be handed the next project, project 2, and if you create a directory project 2 with the subdirectory data and subdirectory results, all the scripts that you wrote for your first project actually copy over to that project's change file families, and the locations will all work. So typically, those of us that do a lot of different projects over time, as I said, I've been doing this 25 years, will have a data directory structure that's basically the same for every project. So I always have a directory with data in it, and then a subdirectory called scripts. And scripts is where I put the analysis scripts, and data is where I put the raw data. And if you open up any project file folder on my computer, you'll find essentially that division of labor. The results are not always in the results. I haven't. It depends on the type of project there. And so that kind of consistency is kind of nice, especially if you're responsible for training undergraduates, because then you can actually figure out, for graduate students, in my case, then you can figure out what they did and where it is, as opposed to if you ever like open the hard drive and like, I have no idea what's where. So I'm a little concerned about you sitting right next to the camera.
Oh, sure, I can move.
Just because if you knock it, then, yeah. All right, sorry. And you can't sit one down because we have a cable there. You could sit in the front row, because then I'd be able to see you. Sorry. Okay. So I'm not going to talk too much more about this. Instead, I want to give you a little Quizlet. Okay. So if you look at the file, if you look at this README that you have, at the top there is there's a little note: in-class Quizlets will be answered here. And if you click on that, you'll come to a multiple choice questionnaire, and I would like you to answer this question by choosing amongst those multiple choices. So, like, just take a minute to answer this, and if you're sitting next to somebody, you can talk to them about it and convert them to answer. Yeah. So I'll just leave it. So anybody have any trouble getting to the questionnaire? It's just at the top of the. So you don't have to answer it. This is not a graded exam. But you can also like bet and not answer, and then that. Okay, so the question is: suppose you have a directory home user work has 3 subdirectories: data, analyses, and figures. What is the path starting with / the full path, to a file output.png that's in the figures directory? There's 4 options. All right, you know, if you answered or given up or find you guys are no longer thinking about it, can you just put up a blue stick? So right at the top of the page of the shell genomics page, there should be a little note that says, right here. Got it? Yeah.
Okay.
Okay. Okay, now I have to figure out how to go find the results. Summary of responses. All right, everybody. I'm going to go for it. All right, so 10 people answered. 1 said option 1, and 9 said option 4. So what was option 1?
That was me.
Oh, you clicked the wrong one? Okay, so option 4, I think is the right one. So home user work, subdirectory figures. So home user work, subdirectory figures, file.pdb. Yep, great, awesome. But since I took so long at the beginning, I'm just going to add some more Quizlets, but I can move on to wildcards instead of doing more Quizlets. So I'll just do that. Okay. It's like the in Cell, I'm telling an anecdote. It's like the faculty. You talk to faculty about teaching, moving away from a lecture format to an active learning format, for example, and they say, and the #1 response you get is something along the lines of, but if we move to an active learning format, we won't get through as much material. In the lecture format, I can really just deliver material. And the obvious response rarely goes over well. Just like, what use is it delivering 2 times as much material? You know, less than half as much material is actually, or a quarter as much material is actually understood. Anyway, so all right. So there's all sorts of like tricks to save time, and we've already done some of them. So CD just like goes to your home directory. And you can also use, if you need to explicitly refer to your home directory, for example, if you want to refer to something that's under your home directory, you can use tilde. This can be hard to find on some keyboards, especially if you have a non-English keyboard. On the Mac, on US Macs and Windows computers, I think it's in the upper left above the backquote. And if you type it, what you'll get is you'll do, if you do CD tilde, you get to the same place as if you type CD, which is your home directory. But the convenient thing about tilde is you can use it to explicitly refer to things. So you can do like ls_data, and wherever you are, that's now an absolute path because the tilde expands to something that's got a slash behind it. Wherever you are, that refers to the subdirectory data under your home directory. And so if you're working on a computer with many different directories, and you just want to go refer to a script or something that you have in your home directory or relative to your home directory, now let's just use tilde/. We've already talked about ... which refers to the directory above your current directory. You can chain it by doing ... /. You can do CD data and then ls dot dot slash dot dot slash dot dot, which actually gives the root of my computer, which is slash. It's the same as doing an ls of slash. Basically, what we're telling you here is that there's a bunch of shortcuts that all do the same thing. And you don't need to remember these shortcuts. This is sort of the way I recommend using information like this is just shove it in the back of your mind, like there's these shortcuts. And then at some point, either someone will use one in a script, and you're like, I don't know what that shortcut is, or you'll come back to this material for something else, and you'll realize that you actually use the full path to your home directory all the time, and you could save a lot of time with it. So these are unnecessary, but helpful. So let's take a look at some actual files. So we've been walking around directories and stuff, and we can do that a lot, but let's take a look at some actual files. And this is something we try to pick an example that would work for people doing stuff. Let's suppose we did an experiment and we're looking at bacterial communities of mice in 2 treatments using 16S. Don't worry about the details, you know what that is, but basically a bunch of sequencing data sets. So you have 10 mice in one treatment and 9 in another. And we also sequence the mock community, so we just check all of our data. So we have 20 samples. We have 10 samples plus 9 samples plus 1. And we've done paired end MySeq sequencing. So we have now 40 files, 1 for each end of the MySeq. And we give them all back as FAST files. And we stick them in a folder called MySeq, which hopefully you've downloaded. And this is what if you're in the data directory, and you look here in ls, you'll see the MySeq directory. If you type ls MySeq, you'll see a lot of files. So these like this. And they're all named, you have samples named 143, 144, 145, 146, not very helpful. The mock community, you probably guess there is the mock community data set. So how do we work with this many files? Can everybody see if they type ls MySeq? Can they all see it's in the data directory? No?
No.
But I think it's the key if you know. Okay, so type CD data. And now try ls. Okay, there's another trick that
I have a question.
Sure, yeah.
When you type MySeq, from where are we getting this?
So when you downloaded that shell data zip, they were in that. So you downloaded everything out of the data directory comes from the shell zip file. So something else I've been doing, and I do this is the problem with, anyway, it's hard to teach some stuff explicitly because there's so much to teach. If you hit up arrow, you'll actually see all your previous commands. And if you hit left arrow, you can actually go back and forth and edit things using, if you type something, and you can insert or delete characters. Okay. So let's navigate to the data MySeq directory. So how do we do that? So ls MySeq shows us the directory. How do we change to that as a working directory? CD MySeq. Okay. And now if you type pwd, you'll see that you're in the data /MySeq subdirectory. Okay. And let's do ls again. And look at that. All the files that you got with ls. Note that they scroll off the screen. There's a bunch there. So another shortcut, and this is actually one that you'll use a lot, is what's called a wildcard. So suppose we want to do things to some subsets of these files. For example, if you wanted to move, so you'll note that the file names are named somewhat cleverly. So they're named with the sample at the beginning, F3D150, And so there's a pair of R1 and R2, both on the same example, and that's because they're parent files. And so you should actually, so the question is there an enabler? Why would they enable this? Question, the answer is because you can do things like file cards that immediately produce files. So I can do ls star, and what star says is match everything. So ls star is the same as doing ls. But if I now do ls f3d150 star, what that's going to do is it's going to match all the file names that start with f3d150. In this case, that's going to be these two. So if you want to do things as subsets of files, it helps to have the files named in such a way that related files, in this case, the R1 and the R2 files, and sequencing are together. So you can put the star anywhere. So if you type ls star fastq, what do you think that's going to do?
It's going to pull out all files that end in fastq.
End in fastq, right? Okay, here's a trick. Here's a question. What happens if I do ls star fastq star? Does it match everything? Does it match the files with fastq in the middle? Does it match things with fastq in the middle, at beginning and ending? Any thoughts? Okay, so don't think too hard about it, because what's the right way to answer this? Steps. People mumble in hopes that I wouldn't be able to identify what they were saying.
Try it.
Try it. Try it. You said try it. You said try it.
Excellent.
Excellent.
Yes.
Yes.
Try it.
Try it. Okay. Okay. Very important point. LS. Never gonna mess you up. Just listing things.
Do you want to see
what
what
a file does, or what a directory path is, or whatever? You can all just say LS. So if you're going to do ls star fastq, and what you'll find is that if you're a computer geek, technically what's going on is star matches everything the empty string, which is a fancy way of saying even if the thing ends with fastq and there's nothing after q, the star will match that nothing. So you could, for example, say I want all of the first claim, all of the left reads by doing star R1. Or I want all of the right reads by doing star R2. And you can do things like this. Star R1 dot fastq will say anything that has R1 in the middle, and anything either side that ends in the fastq. So this starts to get into the sort of programming mentality where you're trying to figure out exactly how to match a particular set of files by creating a wildcard path. Okay, so again, so if you do ls startup fastq, whoops, that worked, right? Yeah. You should see exactly 20 files because we have 20. If you do star R1 startup fastq, you should see 20 files because there are 20 samples. Okay. Why? What value is there in counting files? Any thoughts? So so we're not looking within the files, so it doesn't tell us how many reads we have within each sample. But close. Okay. Suppose you have. Okay, just look at that. You can actually count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So there's 20. Suppose I do this for R2. Looks like I have the same number. That's good. I didn't lose a data file anywhere. As you go through your analysis, one of the most common errors I make when I'm working with anything more than 5 or 6 files is I lose a file. I forget to put it into a command, and it doesn't get analyzed, it doesn't get trimmed, it doesn't get included in the assembly. At the end of the day, I end up with a transcriptome assembly that, all right, I omitted 20% of the reads by mistake, which is probably not a good thing. So it's always worth just. It's worth, there's a sort of metacognition component where you're going, what are the mistakes that I'm likely to make? And you won't know that the first 5 times you do an analysis. But you should be thinking, as you go through your analysis, how might I mess this up? And one thing you can do is you just count the files. Okay. So the way the star thing actually works, and it's sort of a technically important point, maybe not too relevant, but how does this actually work? When the shell LC worker contains the star character, it basically looks in the directory and expands that star to all of the files in the directory that match. And so it actually literally goes in and replaces that star R2 fastq with all of those files. The reason that's important is there's one reason that's very important. LS isn't interpreting the star. The LS command never sees the star. The Unix shell is interpreting the star. Why might that be important? So the reason it's important is it doesn't matter what program you're using, star works with all of them. Because it's not a property of the program, it's a property of how you're executing the program, which is the hash. So if you have an assembler, I don't know, if you have an assembler or a quality trimmer or something that can take multiple files, you can use the star to feed multiple files into it. So that's one reason. So there's sort of this distinction, like there's all these programs that do very specific things with specific flags, yada yada yada. And you have to remember the ones that you use or write them down or something. But star is actually a completely generic component of the Unix shell. So it's really handy to have. Okay. And there's other wildcard tricks that I'm not showing you, like there's a question mark which matches one character and other things, but star is what I use far and above everything else. So let's so I'm gonna do a couple of Quizlets on this. So hang on one sec. Let me come here and clear responses. And so now suppose, so go back to, sorry, I've just completely lost track of where I am here. So now go back to that multiple choice and see if you can address this question. Suppose you're going to measure 3 files, named readme.txt, data1.txt, and data2.txt. What does the command ls space readme.txt print out? So I'll just put up a blue sticky when you're no longer answering the question. If you want to hit it, one thing I'll say is, remember, whenever you have a question about what something's going to do, ls is a safe command to just run. So this is actually a question where if you figure out how to translate something that you can run at your command line, you can actually test what's going on. All right. Okay. So let me just see what the results are. Excellent. We have a divergence of opinion. Okay. So the most popular choice is option 2. The second most popular choice is option 3, and the third most popular choice is option 1, and poor option 4 is left out of the poll. So let's take a look at option 1. So option 1 was ls readme.txt would print out all of the files in the directory. So let's just try it. So suppose I'm in a directory and I'm actually going to cheat. I'm going to go to a different directory, and there's a different file name. So you couldn't just run this to see what it would print out. You have to translate it. And so if you do ls readme.md, it prints out just readme.md. Why is this behavior sensible? It seems pretty stupid if you're doing list files and you're telling it the file you want it to list. And it's telling you, yes, the file is there, right? Very circular. So what use is this behavior?
Check the file is there.
That actually is, yes. So the one thing is like, hey, is there actually a file named readme.md there, right? If you typed readme.txt, it would say it's not there. But this is where you can apply teacher meta-analysis, and we were just talking about wildcards. So it turns out that this is a great way to test wildcards, because you can say, well, I want all of the files that start with R. And if you remember what I told you about how wildcards work, R star is actually expanded by the shell into a list of file names. So this is a way of getting LX to tell you how a wildcard, what files a wildcard actually matches. You could actually do it other ways, but this is nice. The other thing that's really nice is you can do ls minus l r star, and now you're saying, give me the file sizes of all the files that start with R. Which starts to get kind of handy. And I'll show you one specific genomics aspect of this. If you go into the myseq directory and you do ls minus l star r1 star, what do you notice about the file sizes here? Okay, so the command I ran was ls minus l r1 star. Okay, so what do you notice about the file sizes? Is how I know I've been doing it too long, because it's like the first thing I notice when I look at a directory listing. They're all approximately the same. Why would that be? Probably because each file has roughly the same number of reads in it. Okay. That's not so interesting, because there are lots of reasons why one sample might have more data and another sample might have less data, just due to randomness. But what's really interesting is if you then do this. So what I just ran was ls minus lr2 star. And now you see that these files also have approximately the same amount of data. Just to show you what the actual comparison you want to do is something like this, ls minus l mock star. Okay. If you think about the data sets you have, if you have inherited reads, the R1 file and the R2 file should be approximately the same size. If they're not, you've lost some data somewhere. And that's for those who aren't genomics folk, or sequencing folk, that's because the way this works. Like you're supposed to have the same amount of data for both ends of the paradigm sequencing run, basically. Like Illumina basically guarantees. That's the way Illumina works. So this is the specific file sizes. In fact, I can do this because which pair of files are specific to genomics. But you'll often get into a situation. Let me give you another example. I take that file and I trim some adapters off of the file. And then I compare the file size of the file before I trim the adapters off and the output file after I trim the adapters off. Which should be bigger? Before, right? If it's not, maybe there's a problem. So there's all these gut checks. You're like, okay, did that command work? One thing that's really frustrating about computers in general. So I once heard, how many of your grad students? Okay, I wasn't about to install like all grad students. So you don't do that. Well, so the joke is, imagine that the computer is a well-trained but slightly dim-witted undergraduate. They're going to do exactly what you tell them to, and they're not going to know if what you told them was the right thing to do or not. They're just going to do it. And you're not going to know if their results are something you can trust or not. So you sort of want to just like, okay, I told it to do this, it said it did this, and Unix usually will be like, I did that. And I'm like, well, what did you actually do? And this is where just looking at the files, and looking at the file sizes, and looking at the file names, and going, okay, is this working? And again, way too much to remember your first time through an analysis, but there's a lot of things that everybody I know that works this way has converged on, and one of them is like double check after each step. It's sort of like running a vanilla each time after each step of a new molecular biology protocol. Like after you've done it 10 times, you'll know which step usually fails, and you run it till then. But before then, you just double check everything you're doing. And then the fun bits are when something messes up in an area where it's never messed up before, and then you have to like discard all your prior assumptions about where the steps, like the mistakes likely to be. I've spent incredibly frustrating hours in front of the computer going, but that step always works. How could it have failed this time? Anyway, okay, so let's do another quick one. Let me just go back here. Where is it? Dang it. Include all responses. Okay, so the next question, let's just do one more. Suppose you're in a directory with 3 files named readme.txt, data1.csv, and data2.csv, and now you've done the same directory setup. And now let's just command ls start.csv print out. Fill in the form and click submit.
By the way, for those of you that teach with a laptop, this Google Forms trick is actually like I learned it a year ago. It's like super. I never wanted to do quizzes, questionnaires like this in class because I was like, to make a new form and I have to type in the questions. Like, no. You have one form. Just delete the responses again and again and again, and then it gives you the nice bar chart. The other trick was you can have a form where people fill in 3 text responses, and then Google Forms will actually give you, I think it's will give you a word cloud. I think it might give you a word cloud of the words and the responses. It's pretty cool. So I had a class where we had like 80 people in the class, and I was asking them for suggestions on what to do. And they got together a little group for some software development stuff. They got together little groups and they typed out their suggestions. So it was anonymous, and these were undergraduates. Some of them was like, jump off, go take all the here. And the dominant answers were good. So, okay, so let's take a quick look. And I think the real problem with all this technology is that I guess I didn't delete. Well, I'm going to take. I'm going to guess option 4 was the new one. Is that what everybody picked? Option 4 mostly. Okay. Sorry, I thought I deleted it. But so option 4 was data1.csv and data2.csv. Exactly. Yes. So ls start.csv. Okay. So I'll show you what would have happened if there weren't any CSV files in there. But if there are CSV files that end with .csv, ls start.csv will expand to ls space data1.csv space data2.csv, which will print out number 4. And if there's nothing for it that it matches, if you do ls star.foo, it will say no such file or directory. So it'll tell you. So indeed, it's a great way to sort of see what your wildcard are. All right, any questions at this point on the wildcard thing? Okay, let's. I'm going to move on. So we've done all this meta stuff, right? It's like we know how to switch directories, we know how to run programs like cd and ls, wd. We know how to look at the contents of directories, but we actually want to look at the contents of files, right? Files contain our data. And this is where we could do things like look at the number of sequences that's in a file. So let's just start by typing that. Okay, so before you type it, okay, don't type it, first of all. Don't type it. How should we enter that? Any thoughts? How should we type that into the command line without actually typing it?
So tab completion would be one way. Another way that I highly recommend for this particular one is copy paste. Right. So a Unix trick, and again, I've been doing this for 25 years, is type as little as possible. Because every time you type something in, you're going to get it, you're on the risk of getting it wrong. So I use a vast array of tricks, including copying and pasting and tab completion, to avoid typing things like this. You'll also find that your typing speed improves dramatically after using Unix for a couple of months. Okay, so if you enter this cat command, and you paste it, you see this. Anybody know what this is? Well, I'll give you a bit. So the file that we did, doesn't really matter which file, it was S188. If I go here, and I go to my data, and I go to my MySQL directory, and I go in and I find it was S188 here. And I'm going to open this with other, and I think text edit. Text edit. There we are. I can open it with text edit. This is actually the textual content of this file that we just saw. But we saw it at the command line and not in an editor. We used the command cat. Cat is a weird word that I've never run across. It stands for cat-mate. Either they spoke a different version of the English language in the 60s, or these were engineers, or both. And so cat-mate is like take the file and just output it to the terminal. And so what you're seeing here is actually the very end of the file, which ends with ESB dot. And if you scroll all the way down to the bottom here, you see, if you see all the way to the bottom there, it's actually got the dot on it. Okay, so one thing to note, this was a long file. I don't know if you saw it. We didn't have any, there's no pagination on this. When you hit cat, it just outputs the file. And there's reasons for that, and there's ways to get around that with commands like more, which will paginate the file. But it's just showing you the entire file. So one thing, if it's showing you the entire file, you don't want to do this for big files. Generally, you don't want to do this for FAST files. Just an example. Okay. So this is FAST file. Anybody seen this before? Okay. So this is a file that was produced by the UNIX sequencer. They don't have a laser pointer or a stick or anything. So I'll just have to start going. So you see the at M00967 right in the middle. I guess I can use my mouse, but anyway. See that is the line starting with an at. That's the name of the sequence. And then there's DNA sequence, which is actually all one line. The TACGTA line that's just up under that, that's the sequence line. And this is on my sequence, so it's like 350 bases of DNA. Then there's a plus, and then there's the quality score, which is the Illumina sequencer's estimate that each base is correct. So, so this is what mapping programs and sequencing programs and programs to analyze data take in. One thing to note, when I resize my text editor window, see how the lines are actually expanding? No, that's because the DNA sequence and the quality score lines are actually one line. There's no new lines. Okay. So this is what raw sequencing data looks like. It looks like a DNA. And there's one really important thing that I want to convey to you. Never, ever open a FASTQ file in an editor, unless you really have to. So why is that?
You accidentally delete things.
You might change it. And why would you want to change this file? There's virtually nothing that you, as a human being, can usefully do to edit this file. There's two reasons for that. One is like as much as DNA, and I don't know anybody. There was one postdoc in my grad lab who likes to look at genomic sequence with a marker and highlight motion finding issues, but nobody I know actually looks at DNA sequence directly. So one thing is like DNA sequence is very hard to interpret just by looking at it. The other is that suppose like, you know what, I need to change to change the name of each sequence at this time. So I'm going to open it up in an editor, change the name. Why is that a bad idea? Any ideas?
Because it doesn't look like the name.
So one reason is because you have to do it consistently and the other things, right? But there's a less intelligent, but better, I would argue better reason also. How big is this file? Are you really going to change the name of every sequence in this file by hand? Just say no.
No.
Just say no. This is what drives biologists to program, to learn to program. Because you get files with thousands or hundreds of thousands of sequences in them. And your advisor, who generally is a well-meaning but computationally incompetent person, goes, oh, well, it's just easy. We just need to rename everything this. And to them, it is easy because they just told you to figure out how to do it. But in practice, it's actually better to spend 2 days coming over to my lab and learning how to, like, getting a program from us to do it and then learning how to change the program so that it works for you, than it is to do it by hand. For a couple of reasons. One is that after the first time you do it, you will surely need to do it again. And the other is you're going to get it wrong. You're going to go through and you're going to like put new names in and you're just going to get something wrong. And it's going to be a disaster. And in 3 years, when the reviewers get back to you about some weird bit on the curve in your RNA-seq experiment, and your advisor asks you to track it down, and you're already on your third, your third career move after that lab, and then you're going to find out there's a typo, and some of your data steps aren't validated. It's just a nightmare. And odds are pretty good what you're going to do is Photoshop a little bit out of the graph. I know you think. I mean, on the one hand, it is kind of funny. On the other hand, like this happens. So we're trying to get you off on the right foot. Which is just say no to editing things like this manually. Never load these files into spreadsheets and save them. Never save these files. You can look at them if you really must, but never save them. Always use a program to manipulate them. Most programs will have checks to make sure that what they're doing is consistent. The flip side is the programs will mess these things up on a grand scale, right? So editing this is going to be annoying, and you're probably only going to make 1 or 2 mistakes. With a program, you can make mistakes on a massive scale. So there's the flip side there. So there's no free lunch here, but the programs are going to be the standard way of working with this. I don't know if you can tell, I spend a lot of time thinking about sequencing data analysis. I spend a lot of time thinking about this. Okay, so we want to make sure. Okay, so we're going to do some stuff. So make sure we're in the MySeq directory. So you should be able to do that with cd/data/MySeq. If that doesn't work, please put up a sticky. I'm going to do cd/data/MySeq. And if you type pwd, you should see that you're in MySeq directory, okay? So for these big files, so there is a reason to actually look at these files. And the reason to look at these files is to make sure they're actually what you think they are. And this may sound a little counterintuitive, but it's like, hey, is this a FASTQ file? I don't know. Is it full of gobbledygook that looks like the DNA sequence with @ signs and stuff? Yes, it's a FASTQ file. You'd be surprised how often file names get messed up. The sequencing center may just send you the wrong thing, for example. Right? It might not be a FASTQ file. Or your undergraduate student that's working with you may have messed up the files, all of the files, and you're wondering why none of the bioinformatics students are trying to run work on them. Or you may have messed up all the files and you're wondering. And so just being able to look at the file is your first line of defense, right? It's like, it's like look under the microscope when you're working with an organism. Like maybe it's dead. As I said in the embryology course, death is a phenotype. Just not that interesting one. Okay, so there's a program called less. So cat is very useful because it gives you the end of the file, basically. And if you're looking at a big file, it's annoying.
So use the command less instead. And less is a program of a type that we haven't encountered before. It's a program that doesn't exist immediately. So before things like CD and ls gave you an answer and went back to the shell program. Less leaves your inside less right now. But less does. It's part of the joke. So there's a command called more, which doesn't say anything, it says not as null. And there's a new command called less, which does the same thing as more, just better. So less is more. UNIX engineers, jokes, humor. Anyway, what less does is it's a tab here. It starts at the top of the file, and it lets you page down by hitting the space bar. Lets you page through the file. So if you hit the space bar, you're walking through the file. It also has all sorts of other useful commands. So suppose you were silly enough. Okay, here's actually a really good reason to do this. So suppose you're like, you know what, my mapper said, try to map sequence M00967:43. Problem, this sequence doesn't look like DNA, failing miserably. This is the kind of error message you'll get out of this program. And then you're like, okay, how do I figure out what sequence that is? Well, what you do is you can copy the sequence name, like so, and then in less, you can type slash, and you paste the sequence name like that. So the slash, and then you can just type in whatever, some character. And what it will do is it will search through the file and find that sequence name for you. Now, this case, I think every sequence in the file starts with that. So you'd probably be better off using like the thing at the end. So suppose you're looking for this sequence, 3345, and you go in and you type slash and then paste, it will actually go through the file and find that. So this is a useful way to find sequences by name, basically, in less. And since I just did a bunch of stuff, basically the three commands I'm using, I'm typing less of the command line, I'm typing space to page down, I'm typing Q to quit out of less and go back to the command line, and I'm typing slash name enter to search. And there is a helpful, everything but slash is here in this helpful, badly formatted table. Oh, and the slash. So this is something that when you get a big data set and you're trying to figure out, you're trying to troubleshoot what's wrong, you can use less. Okay, are there questions at this point? So it's a little bit weird. Like less is a we'll see another example of this with editors that get down to scripts. Less is a program that you run at the command line, that you go into the program and you don't exit until you tell it you're done. And knowing how to get out of programs turns out to be an important thing. And it's different for every program, potentially. Q and escape and so on are useful. Okay. So if you're in less, type Q. If you're at like a prompt like this, type Q, and you should be back to whatever your shell prompt is, which for like is percent, for me is percent. Yeah. Just to show this is going to be the direction. 2% is just useless loading, just tell them shell prompt. If you look at what I configured that for the workshop, what I actually have my prompt do is, this is what my default prompt is. I have a terminal that's got a computer name, Siasa is the name of my laptop. It's got a tilde, which is the directory name, and then it's got the prompt. So if I do like cd/tmp, in my prompt it tells me what working directory I'm in. In fact, we should probably just put that in so like configure your prompt to tell you what directory you're in. And so if I do like cd/smart, the problem with this is that sometimes my prompt wraps, I'm in a deep enough directory. Sometimes like my prompt is like super long. And that gets kind of confusing. But so you can do all sorts of things to configure your prompt. And if you find yourself working on multiple computers, it's helpful to have a computer name of the computer you're working on in your prompt. So you can have a terminal window open on your laptop, and a terminal window open on remote computer 1, and a terminal window open on remote computer 2. And someone you forget whether you're working on a computer in North America or in China. So this is actually a really good time for another digression, which is you are now very well positioned to look to watch a certain class of movie. Where is it? So there's actually a Tumblr where you can see people typing in Unix command line things like, so this is code. But so I don't know if you saw like Trinity in Matrix shutting down power plant Unix shell SSH. Oh, no, sorry. Okay, black button is optional. So what she's doing there at the command line, this is where she's shutting down the power plant. I think it's like this third line. What she's doing is she's actually logging in and shutting down the power plant remotely out of Unix terminal. And you can actually understand some of the commands. And apparently the commands are legit. Like, people have gone and analyzed them. And then I think, if I remember right, it's the line where immediately after this, she then rides a motorcycle full of explosives off a roof, backflips off it, and like completely blows up the guard shaft. But anyway, so that's all of those things are optional when you're working with the command line. But you will see you'll see this in movies now where you're like, oh, they're typing something. You're like, there's an LS. And there's also the ones where you can actually, there's websites where you can go and you can just do the cat walking on a keyboard thing, and it'll like output streams of legitimately text, so you can press your friends by pretending that. Anyway, you probably don't want to try using this to impress your friends, just to be honest. But yeah, okay. So we looked at less for looking files. There's also two commands, head and tail. And head and tail are really useful for like just quickly looking at a file. So less is if you want to like search a file or page through a file. Head and tail will show you the beginning and end of a file. So why might you want to look at the beginning of a file? Let's go with a sequencing paradigm. Any? You run a mapper, and the mapper says, this doesn't look like a sequencing file. What's the first thing you should do? Look at the file. Okay. So suppose you run the command head. What head will do is show you just the first bunch of things in the file. So I just ran this command. And this is useful for figuring out if a file is even remotely what you thought it was. Like if it doesn't look like this, it's not a FASTQ file. Okay, so that's why you might want to look at the beginning of the file. Why might you want to look at the end of the file? I'm realizing as I'm saying this how deep my computational paranoia runs. Because the answer is, what if you really don't trust the file? Well, you look at the beginning, you're like, okay, well, the beginning's fine, but what if it's messed up at the end? And then you go look at the end. And if it's good at the beginning and it's good at the end, then odds are that it probably, if somebody messed up the file, it was a human being editing it. Because computer programs are going to trash the large sections of the file, and they're not going to be discriminate, so it's probably going to break at the beginning or the end of the file. You're going to see it at the beginning or the end of the file. So head and tail are great ways to just gut check down, like, is this a sequencing file or not? So, you know, some people call it paranoia, I call it experience. And so it's fun, you can actually say, I just want to look at the first line of the file, or I want to look at the last line of the file, and I'm just going to copy both these commands and paste them in. And so you can see the first line of the FASTQ file is the name of the sequence, and the last line, the FASTQ file should, the well-formatted FASTQ file should be the quality score, because each record in the FASTQ file starts with sequence. So, searching files. So we showed you how to search within a file using the command less. There's another command called the grep. I have no idea where the word grep comes from. I'll look it up later.
And what grep will let you do is it will let you search multiple files for lines matching string. So suppose I want to know which file contains 1101:14341. And here I'm just going to search in a specific file. I do that, and what grep does is it returns the line that contains that substring that I searched for. If I search multiple files, it will tell me which file it's in, which files it's in. So I can do that grep, and I can do start fastq, and it will tell me that it's actually in 4 files. Sorry, I know it's a little hard to see the way I've got the colors configured, but here's the command I ran. And what you can see is it's telling me it's in 4 files. It's in R1 and R2 of sample 188, and it's in sample 213 as well. Why might that 1101:14341 be in multiple files? Any ideas? Okay, first of all, look at the sequence names to the right. Are they the same sequence names? And the answer is no. One has 8428. One pair of sequences has 8428 in it, and the other has 26115 in it. So the problem with grep, it's not a problem. The deal with grep is that it looks for substrings. So it's going to find any names that have that 1101:14341. And Illumina, the names aren't random. They actually specify locations in the flow cells and so on. But you might as well assume to be random. And so, it's just finding that sequence is in. So if you wanted to just know which sequence, which file that particular sequence is in, you want to take more of the names you need. So you could actually do, if you wanted to know all of the files that contain the sequence name that match that, you could do grep that in startup fastq. And now you find just the 2 files that have 8428. It's in an exact match installed. And there's ways, we're not going to introduce, but there's ways to put wildcards in grep. So you can say, you know what, give me all the files that have anything that starts with M00967 in them. And with FASTQ files, it's a little hard to give an example of that, but that's actually really useful for looking at adapter sequences. So if you have if you're wondering if there's a barcode that's been left on your sequences, you can actually grep for the barcode. And if you find there's like 100,000 matches, then the barcodes have been left on your sequence. If you find there's like 5 matches, then the barcode has been left on your sequence. Does that make sense? So it's sort of this weird thing with Unix, and it's generally true of programming, where we're giving you the raw ingredients, and you can recombine them into whatever recipe you want to achieve whatever you want to do. And again, hopefully what you'll remember on all of this is that there is a way to do it, and it is in this long document. You can come back to this long document and look it up later. Or you can come to the meet analyze data thing and be like, I can't find, the document's too long, I can't find it in there. Please just give me the answer. We'll give you the answer. So this is actually pretty useful. This grep command is pretty useful if you actually want the whole sequence, because you can say, give me the 3 lines after it using -83. And now what you're getting back is the full FASTQ record. And it's just there's a colon, and then the 3 lines after it have dashes there. And so the full FASTQ record, if you want to know the sequence of that record, just use the mouse. If you wanted to know the sequence of this FASTQ record, then if you use the -A command, you'll get the match, and you'll get the DNA sequence that's after it. This can be pretty handy in situations like in the 16S situation. You may want to know there may be a particular OTU that's created by a particular sequence, and you may want to take that sequence and do like blasting or something more intensive on that DNA sequence. And this could be a way to extract that sequence without opening the file up in an editor. So another thing, to sort of make that point, if you open these files up, at some point the FASTQ files you get are too big to be opened, like Word or Excel or an editor. grep can deal with them just fine. So we haven't really emphasized the scaling aspect, but a lot of these tools work with the files piecemeal. So grep is only going to, grep is never actually going to choke on a file size, as far as I know. It just may take a long time. But you know, you may get too much output from grep, but it's never going to go, oh, this file is too big for me to work with, because it works through the file in a piecemeal way. It doesn't try and load it all in and display it. Okay. So this is the kind of thing you can do, and it's actually really useful already for FastQ for people working with sequencing data sets. And maybe we'll finish, I don't know how long this is. Okay. We're going to stop before we get to scripting. We'll stop at scripting. Scripting is, I'll give you a 5-minute intro to scripting at the end. But let's talk about creating, moving, copying, and removing files. Okay. So files are in directories. We can move around in directories. We can list the files in directories. We can look at the contents of files in directories. What if we want to change the name, rename files, or copy files? So let's start. So you could do this with the drag and drop and the normal thing. Why might you not want to do that in this circumstance? What's an example? Are there any situations where the drag and drop or copy paste is not going to work that well for you?
You are using a remote computer. You don't have a.
Yes. You're using a remote computer. You can't use the terminal. You can't use the file here. Why don't you want to do it on your local computer either? Well, how many FASTQ files do you have? 20 or 40? 40, something like that? So if you want to copy just the FASTQ files, you have to figure out how to sort your file view so that it looks right. And that can be done, right? You can sort by like extension or something. What if you want to copy all the R1 files, but not the R2 files? There's no actual simple way to do that, I think. I guess you could search for R1 and take the results, but I've never actually figured out how to use the max search within a directory thing that well. So this is a situation you can use wildcards. Basically what I'm saying is you can use wildcards, right? You can copy files to a directory using a wildcard, share it in a subset of files if you want. So in this case, we're going to start using the cd command, which is copying using a file. And there's file stability.files, and this contains this is metadata for the sample setup. You can actually look at it with cat if you want to see what's in there. You can do cat stability.files. You can see it tells you, it says the mock. So this is a tab delimited file. It says mock. The mock sample has 2 files associated with it. That's on R1 as the R2 file. And this is basically a recapitulation of the directory listing, but it's in a format that I think Mother, which is a 16S analysis program, will understand. So one of the things you'll find with bioinformatics is every program takes input files in a slightly different format. And you figure out what that file format is. So this file is pretty important because it tells it's something you need. This is important as your raw data because it tells Mother where the data is. And what data goes with what data. So let's back it up. So the way to back it up, and here I'm just going to copy paste, is I'm going to use the command cd to make a copy of stability.files and name that copy stability.files _ backup. If you do that, you'll see that it looks like it succeeded. How do you know that it succeeded? Because it didn't complain. I don't know if this is a good thing or a bad thing, but Unix, a lot of the time, Unix will output a lot of stuff when it is doing something, or it won't, and you can't really tell. It depends on the command. CP is one of the commands that's strong and silent. Like if it works, it's silent. If it fails, it'll tell you I couldn't do that. Disk's too full, or you don't have write permissions, or that file already exists, or whatever. And now, if you do an ls of stability star, you'll see we have stability.batch, stability.files, and stability.files _ backup, which is the one we just created. And you will, of course, also see that
here in the directory listing, where we now have stability files.backup, which was actually modified and created today. Okay, so this is all well and good for copying files, but for renaming, for making a copy of files, but we also suppose we want to make a backup copy of a bunch of files, and we want to put them all in another folder. So this is where a command called mkdir comes in handy. This is a way of creating a folder after the command line called make directory. So if you type make directory backup, now what you will see in your file viewer is the directory backup has now been, the folder backup now has been created, and it's empty because you just created an empty directory with mkdir, and now you can move the file stability.files_backup into backup. And now when you go look in your file viewer, you'll see that the backup directory contains stability.files_backup, and life is good. Good so far? All right.
So what's the slash? Why is there a slash there?
To tell you back as a folder.
It says backup as a folder. Yeah. So you don't need it there. If you say mv stability.files_backup space backup without a slash, it will figure out that backup is a folder and put the file in there. The problem is if the folder backup doesn't exist, what mv will do is rename this file to be named backup. So if you have a typo, hypothetically, this is paranoia at work again. If you have a typo in your directory name, this happens to me like once a day. I have a typo in what I'm moving the file to, and it renames the file rather than putting it in the directory. And then I have this really annoying thing where I need to unrename the file before moving it into the directory. If backup doesn't exist, this actually will give you an error because and I'll just show you that, you don't have to follow along. But if I make another copy, and then I do this mv, and I say mv stability.files backup // it will say no such file or directory // If I take off that slash, it thinks I want to rename the file, and it will succeed just fine. Very common error. This is a fine example of Unix being like, oh, you have a loaded gun and it's pointing at your foot. Please go ahead and pull the trigger, right? Like, it lets you shoot yourself in the foot. And the slash is like a safety. You should probably come up with a better analogy than loaded guns these days, but anyway, habits. Okay. So, okay. So this is like, let's continue using this for file. We're going to rename it underscore important. I don't really know why. You would never actually do this, but let's just rename it. So now you can see if you type mv, this just renames the file, and so now there's a file mv. There's a file stability.files_important. Okay. And just to continue whacking this profile, we can actually remove files also. So, and the remove command is rm. If you do rm backup/stability.files.backup, it will get rid of, backup, rm backup / wait, what do we want to do? Yes, we want to do remove backup stability.files.backup. I use tab completion there. I just type B and then tab, and it would complete backup, and then I hit tab again. There's only one file in backup, so I'll automatically figure it out. Okay, so if I hit enter, it goes away. And here's the important thing about rm. rm, the files are gone. There's effectively no way to get it back. It doesn't put it in trash. Depending on how, sometimes it will ask you to erase short, depending on how your account is configured. But it's gone. Once you say yes, it's gone. You can get it back. So just keep that in mind. Okay.
So let me see if I have had one more. Do I have one more Quizlet? Nope, no more Quizlets. Huh, I wonder where that went. Okay. So, so at this point, are there any? Where did they go? What's happening here? Okay. Are there any questions on copy, MB, RN? All good-ish? Okay. Let's see what else I tell you about. Let me tell you one more useful command. I should probably put it in this tutorial. CP minus R will make a recursive copy of something. So suppose that I am in the directory above MyC. So what I do is you can do it here by just doing cd /data, and it should bring you to here. Suppose I would say, you know, I'm going to be working with MySQL. I want to make a copy here or on another hard drive or make a copy. But I don't want to go into MyC and copy each file individually. You can use wildcards with CP, or you can go find the directory that contains everything you want a copy of, and say copy minus R, MySeq to MySeq.back. If you don't put in the minus R, what it will tell you is I can't copy a directory. If you put in the minus R, magically it can copy a directory. And now MySeq.back contains a complete copy of MySeq. Why? Any thoughts as to why Byte doesn't do a copy minus R by default? It's actually not because it's trying to be friendly to you, necessarily. I mean, Unix isn't friendly unless you're trying to do it. So imagine that you do copy minus R on like your home directory. It would be like copying your home directory and pasting it somewhere else. It would take a very long time. So there's I don't know that this is a general statement that I can make, but I'm going to make it anyway. Generally, Unix will ask you to the default behavior of Unix will not be to. Is this true? I don't know. The default behavior of copy is not to something to create a lot of new files. So you have to tell it explicitly that you want it to create a bunch of new files. I was going to say generally Unix doesn't let you do things on a large scale, but you can delete as many files as you want on time. So that would be on. Okay, so this is okay. So I guess we have a little bit of time. So let's spend the next little time talking about writing files. So this is probably once you get to the point where you're using ls and cd and all this other stuff, and you're running commands. And which commands you run? I mean, we're not talking at all about what you might actually do in terms of running mathematics workflows. And I do have tutorials and tutorial materials on that use all of these skills. So I can point you at those. But this is really sort of more about the Unix shell than about any particular workflow. The thing you need to find next is you actually want to do several things. You want to run, so again, sequencing again. I want to trim the sequences, I want to map them, I want to take the results of the mapping, and I want to convert it to a data file. Something like 6 commands, 6-step set of commands. And maybe these commands each take half an hour on the data set size that you have. So what you want to do is script them. And scripting will just mean automating them, which really means laying out the commands in advance that you want to apply to. And the way you do that is you write what's called a script file, or a script. And a script is really nothing more than a text file that contains the commands you want to run. And the first command, the second command, the third command, the fourth command, the fifth command, that's it. So what we're going to do now is how to create that script file. We're not going to tell you what to put in it, because that's going to depend on what you want to do. But we're going to show you how to create it. How many of you work on like the ARM cluster or any of the or any of the other campus compute clusters? Okay. So script files are also how they are the same thing as the
as like the QSub files. They have to use QSub, right? On the farm. How do you submit like a batch? They're batch files. They're the same thing as batch files. They just contain the commands you want to run. So this is the same way that you would submit something to a queuing system. But those of us who don't use CPU clusters, we have a compute cluster. Typically, you have one computer where you manipulate files and set up commands. Then there's a bunch of other computers that you don't have direct access to. And what you do is you describe what you want those computers to do in a batch file, which is basically a script file, and then you submit that, and it runs it when there's free space or time on one of the computers. So it's a way of sort of leveraging big compute resources without having to specify which computer you want to use to do something. If you have like 50 computers, you don't really care which computer does it. You just want one of them to do it when it's free to run it. So that's how you actually will do a lot of things on those compute clusters. Is you'll write a little script and then submit that script to the system that decides where to run it. But it all starts with writing the script. So we're going to use the program Nano, which I think is installed on the Windows computers as well as the Mac computers. So if you type nano space awesome.sh, you should get something like this. Does that work for people on Windows? No, Nano doesn't work. Can you try Pico, P-I-C-O? Also, Pico does work. Just take a quick look. It should have been installed, but we probably messed up the install instructions. So yeah, so now, okay, yep, everything's running everything right. So so there's nothing here. So is there a text? Does anybody know what the text editor is on Windows? Is it symbol tab? No, it's not symbol text. It's Notepad, right? Notepad. Okay, so go find Notepad.
Yeah. So the commands should be.
Yeah, so if you can find the Notepad program, I don't know how to find it. Just type Notepad down and ask me anything. There, and run that. Okay, now the important bit here is that you're running these Notepad on Windows. You need to save it into the directory that we're working on. So go to File, go to the File menu, and do Save As. And put it in the, if you can find the directory, which is going to be this PC. And that's not so helpful because it doesn't work. Devices, drives, C, and then users and so on and so forth. Okay. And I hope this will work. I'll check in with you and then go to the data directory. Sorry, I made a note to myself. Okay, so for those of you that are on Mac, this will work. This is actually a fairly standard UNIX editor. This will be on most computers most remote computers that you have access to as well. And that's the difference again between running this in like Notepad or I don't know what text editors do people use on their Mac on their Mac laptops? Anybody? Text Wrangler, right? So you can do this with Text Wrangler too. The difference again is the difference between a local and a remote computer. Nano will work on a remote computer as well as a local computer, whereas Text Wrangler and Notepad no longer work on a local computer. But again, it doesn't matter, or not again, but it doesn't matter how you edit a file. What matters is that you're creating a file that has the instructions in it. And so, okay. So you can type in a command. So if you can go find that grep command that we used up above, it doesn't really matter which command, but let's just do something like grep. You can put that grep command into your Nano editor. It's going to look like this, and Notepad++, same thing. You just copy and paste that line of text into Notepad. And this is now exactly the command that you ran at the command line, but now it's in a file. What does that mean? Well, it just means there's a text file with that command in it.
And if you do Control X and no, it will say, should I save this? And you can say, yes. Enter. I don't know, did anybody see that super awesome thing where you just Control X?
It was like save and then it has the little hint. Answer no will destroy the changes. This is sort of your UNIX hyperbole. So I'm going to say yes.
And then finally to write. Is everybody good with the Control X and then yes and then finally? Okay. And now you'll see that there's a file awesome.sh that wasn't there before, I think. It was there before. You can actually see what's in it by typing cat awesome.sh.
If you're working within Notepad, it shouldn't be as simple as saying save or save as and naming it awesome.sh in that folder. And then if you type cat, you may not see it. Now there's one caveat, which is, so let's, I'm just going to type something below. And if I do save as, so save as, see if it puts in the right, and let's say awesome.sh. And if you go to your, do ls here, you should see awesome.sh. And now try typing cat awesome.sh. See what happens. Okay, remember you can use tab completion as well. And it's in there. Okay. So the key thing is don't use Microsoft Word to do this. And the reason you don't want to use Microsoft Word to do this is because Microsoft Word by default doesn't create text files. It creates Word documents which have a lot of other stuff in it. What you want is this something that outputs text. So anything that's like a programmer's editor, like RStudio or Text Wrangler or Atom or Notepad++, will do this. Notepad will also work. So now that you have this, you don't have to have the grep in there. It doesn't matter what you have in it. Now, how do you run it?
Well, when you're running the bash script, this is interesting. Let's just run it. There's a lot of discussion here. But so you can run it by typing bash awesome.sh.
And I get an error, but it runs. So right now, bash awesome.sh, it's giving me grep, blah, 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 just try that error. So what's going on? Why isn't this working? Okay, first of all, where the hell does grep come from? Why is grep complaining? I ran awesome.sh. It's not a check. Well, if you look at the contents of awesome.sh, awesome.sh contains the command grep. So bash is not being very helpful here. Bash is running awesome.sh, and rather than saying, hey, you have an error in awesome.sh, grep is complaining. It just passes the error grep. So the better question is, why is grep complaining? Any ideas?
Because I'm not in the right directory. Yeah. If I just ran the grep command, it would have the same problem. I'm not in my. So if I go into the my C directory, and now I'm going to try to run awesome.sh, and it's going to fail. Why is it failing now?
Because the script is not there.
Right, because the script's not there. How do I reference the script? With the path, and what's the path? So conveniently, so again, so this is not something that you should have in your short-term memory yet, unless you've been doing this a lot. You see the CD command? I did CD myc, and that was with relative path. So what I did was I was in the directory, and I changed to a subdirectory called myc. And that previous directory was where awesome.sh was. And how do I refer to the enclosing folder with the relative path? I Anybody remember?
//
So this is where, like, you know, it's, I recognize that it's not easy and that this is where a lot of people have the most trouble the first couple of weeks they're using the shell. I knew I was in a directory at all this run and then I did the CD, so I knew I was in a directory below that directory. So I'm keeping the locations of awesome.sh in my seat in the files in my seat in my head. It doesn't mean that I'm a sane person, but I am an effective Unix user. That may be a distinction that becomes clearer to you if you're using Unix for a while. So I can do this. So it becomes a little bit weird. I'm running awesome.sh. I'm running the commands that are served in another directory, but I'm running them in this working directory. So the grep command will operate on the files in this directory, even though the grep command itself is in a file that's somewhere else. All that matters is the commands you're running, not what file they're sitting in. I do that, I get a result, I get my grep result, and life is good. And I show you just very quickly, if I do nano.awesome.sh, and I now say echo done, this demonstrates, or maybe I can do echo, because I'm really interested. Echo just prints up the screen. This really starts to illustrate our script. There's now I have two commands in the script. With a single command, I can run two commands. Right? With bash awesome.sh, I'm actually running both of these commands. So to show you that, if I run bash awesome.sh, it does the grep, and then at the bottom it's the little done. That's what echo done prints out. Okay. So how should you be thinking about scripts? How should you be thinking about writing scripts? The important thing about scripts is that they're close to the right way to write them, the correct way to write them in my experience is the dumbest possible way, which is you run a command, it works, you copy it into like an email to yourself or a notepad document or something. You run another command, it breaks, you fix it, you copy the command, it works. It's good notepad document. And you do that and you build up your workflow. Like these 5 commands, they all work. Eventually got them all work in a row. Then you take what's in the notepad file and you start running commands one after another at the shell prompt. And once that works, because maybe some of them are modifying directories, modifying files or whatever. Now, once they all work, once they all fit together, that's your script. You've recorded what you've done. It's in a script. It is now automated. And here's the really, really cool thing. If you do just that, you just have it all in a script, that's your method. That's part of your method section for your paper. This is what I did. Here you go. And if you include that script in in a paper on what you did for sequence analysis, for example, reviewers like me will be satisfied. Like, okay, I can find out what you did. I mean, we may still not like your paper, but we can at least know what you did. All too often with data analysis, the results you get depend critically on what choices you made in the data analysis, and all too often, people don't tell reviewers or readers what they actually did in their data analysis. If you provide a script, you're like, yeah, you don't have access to my computer, so you don't necessarily run all this yourself. That's a whole separate set of discussion points. But at least you can see, okay, you use VWA, you use this command on VWA and map the reads, or use this sliding monitor. And the reviewers should be expert enough to know whether those were the right commands to use. If they were the right commands to use and you put them in a script, what you're doing is you're signaling that you know what you're doing. And for reviewers, that's actually often enough to be reassured about your sort of base level of influence. So completely apart from the whole, like, can we get things published, it also means that when your advisor comes back to you and says, you know, I think you did single-ended, I don't know, use this reference genome and you should use this reference genome. Or, much more likely, use this reference genome and now a new reference genome's out. Can you rerun everything? You have a script. You edit the script. You rerun everything. You go to the beach for a week. You come back, and you put what's this? Is it foundation that makes you look pale? So that it looks like you haven't been at the beach for the week. So yeah, I spent the whole week rerunning our entire analysis to huge amount of work, here are the results. And then you're done. If you get too quick at this, your advisor will not appreciate it. So always pretend to take longer to run an LCH than you actually do. It doesn't work in my lab, just in case you're wondering. But that's okay. If you get too quick, I have to be like, I know I asked you to do something that requires a week of compute time, and I asked you to do it yesterday. Where are the results? Sort of the attitude. Anyway, you know, you all have a question. So these scripts are like really useful. They record what you did, so you can rerun it again, so you can convey it to other people, so you can run it again on a different data set by editing things, and that's really, I think, where the power of Unix comes to the front. There's no way to do something like this with the graphical user interface, effectively. And that's why, basically, this is why bioinformatics and large-scale data analysis always depends on this command line. There are other approaches to recording the actions that you do, and they all rely on basically, here's the command that I actually want to run. So if you use like RStudio or Jupyter Notebook, there are ways to do it in those as well. And so, are there any questions at this point? Yes, please.
How did you edit the awesome.sh? Can you show me it?
So I ran the same nano editor or Notepad or whatever, and I just gave half to the script, and it loads what's already in there for me to edit, and now I can edit it. And now it's done. Sorry, I do these things too fast.
So I don't have the word done. It says my output. Like,
That's because I put it in. That was what this. That's if you go and edit file nano and you put echo done in, then yeah. So I put it in explicitly to show that you could change multiple.
What was the command to save it again?
So another Unix step. Whenever you see the caret, they usually use the control key, so control X.
Okay.
Good-ish. Yes. There's a lot of tricks to writing scripts, and then there's like programming you can do in scripts to do the same things that I run files, and that's a little bit of an argument. One caveat, one warning in scripts, and this is something I learned a year and a half ago. One of my grad students was like, "You're writing scripts wrong. Write them better." Is the commands in the script will get executed whether or not they fail. So the second command, the script won't exit if the first command fails. It will run the second one. So just to illustrate that, yeah. So suppose right now in the MySeq directory, so that grep command works. If I change back to the directory of the MySeq directory, to where we get this error, and I type bash awesome.sh, and now I get this error, you can see that the echo, even though the grep fails, the echo still prints out the test. That's bad, because if you have like 100 commands in the first 1 minute, probably the rest shouldn't run. So there's probably, I don't know why it works that way, but there's two, and I never remember which one does which. There's two things you can do to modify the behavior of bash, to then modify the shell behavior, which is set minus x and set minus c. What these will do, one of them says, print out your command you're running, and the other one says, if the command fails, this is the reverse. And you just put them at the beginning. So if I save that, and now I run it with bash, what you'll see is two things. The commands prints out the commands that are being run by bash are printed out. I think that's what set minus x does. And then the grep command fails, so that echo is not printed out. So this is so that the set minus x and set minus c are things that should be in every bash script, unless you want it to continue going after it if the command fails. It can actually be kind of annoying because sometimes if you have like a remove command, and the remove command will fail if it doesn't remove any files. So you're going to clean up after command, you're cleaning up the directory, and there's nothing to clean up. The script will fail, and there's ways of getting past that. But in general, it's sort of a good default to have the set minus x and set minus. All right. So I think this is probably an okay stopping point. We are more than happy to have you come to office hours or send us an email. I'm more than happy to have you send me an email about stuff. And the reason I say that, just to tell you why I'm more than happy, nobody ever sends me an email about this stuff. Okay? Basically, what happens is people come to these and then they disappear. So, you know, if it gets to the point where all of you are sending me emails, I may be like, no, I don't want any more emails. But right now, intuition suggests that there's some barrier, like you think your question's not good enough for me to answer or something. Lower the barrier. It's okay. Ask me the question, or come to the Meet and Analyze Data session, where we're friendly. We have coffee. Sometimes we'll have donuts, and you can ask all the questions you want without fear of them being viewed as unique. Too simple. The goal is for you to analyze your data, not to look smart while not analyzing your data. So I'll just say, just to sort of go back to the beginning point, this is hard stuff. Nobody is good at it naturally. I have literally been doing this since 1988. Kid you not. Kid you not. Not data analysis, not bioinformatics data analysis, but like Unix. You know, there's no like. Oh well, that person is that. I would never do that. So I'm just going to give up. None of that actually is true. It's just the person that looks good and didn't do it for a long time, basically. So this should give you a little bit more insight into what some of these other things are. I haven't actually specifically talked about some of this stuff. So spreadsheets, the big problem with spreadsheets is you can't automate the things you do with spreadsheets, right? Like if you go in and you fix a spreadsheet, like there's no way to apply that fix across all of the rows of the spreadsheet. What OpenRefine does is give you a way to do that. OpenRefine is a way of doing large-scale manipulations to spreadsheets. It's like it's mind-blowingly awesome. If you work with large, complicated spreadsheets or large, like row tables spreadsheets, OpenRefine. The comments we get back after demonstrating OpenRefine are like, I would have saved 2 years of my life if I'd known about this medication, that kind of thing. Like it is ridiculously awesome. And we'll also talk a little bit about spreadsheets and how to use spreadsheets better. There's some ways to sort of do things to spreadsheets to make it less likely that you're going to make a mistake. Everybody knows about the date problem sequence, okay? Sequence analysis. So in spreadsheets, if you annotate your genes, the BLAST and stuff, you get a spreadsheet with all your genes annotated. Excel will interpret the names of genes. So OCT1, which is a common gene or stem cells, I believe, or OCT1 or OCT4. Anyway, stem cell gene will actually be turned into October in your spreadsheet. There's ways to avoid that. Somebody actually did a meta-analysis, published a literature, showed this was like an epidemic problem. Introductory version control kit. So something that there are a bunch of problems that you will start to appreciate after you start using the stuff that we talked about today. At some point, you're going to look at you're going to have a bunch of scripts. You're going to have done a bunch of data analysis. You're going to have like 10, 15, 20, 30 scripts in your directory. You're going to be happy as a clam. And it's going to be wonderful. And then at some point, you're going to have the script that you ran last week, and the script that you ran 6 months ago, and the script that you used to analyze the data 12 months ago. And they're all going to be different versions of the same script, and they're going to be named something horrible like script.sh, do analyze data.sh, do analyze data _ try advisors advice.sh, try advisors advice 2.sh, right? And you're going to run into this problem, you don't actually know what you ran. Right? And then what version control does is let you recover old versions. It's like over change tracking, but you recover old versions of scripts. And version control is probably it's one of the like top 10 things that I've ever learned in my life that's changed the world. So for me, version control Git is sort of confusing, but it's what everybody uses. So if you've heard of or you're moving to another university that doesn't have access to a powerful computer, you can't run a lot of analyses on your laptop. You need sometimes more memory or more disk space. With Amazon will let you rent a computer by the hour. Amazon and Google Cloud and Rackspace and others will let you rent computers by the hour with more disk space and more memory. They'll let you rent Windows computers or Unix computers, so you can run those analyses. All you need is and so we'll show you how to set that up, log in remotely, and run those things up. That's actually where we do. My lab does a lot of data analysis, because sometimes if we have 100 new data sets, we can actually analyze them all at the same time by renting 100 computers rather than trying to find computers on things. Yeah, and that's it. I'm trying to figure out what to do for these to be determined dates. I'm thinking that I might do, for one of them, I might do an intro to like data notebooks. So you've heard of RStudio and an overview. So this experience. The question is if I do like boring work-related, like directly work-related things, it's too close to Christmas, so I don't want to think about work. Whereas if I do like shiny new technology, people might show up. So I have to think about it. So if there are topics that you're interested in, you've heard are cool, and that you want me to teach you for half an hour, for 3 hours, then drop me a note. I could also do transcriptome assembly, like the sequence trimming and transcriptome assembly. And I think that's all I have to say. Any questions on? Or is it like blood sugar low, coffee needed, or lunch needed? Great. Thank you all for coming. Please interact with me or my lab about these things. We're here because we want to increase the community that does this on the campus. And maybe I'll see you in the Thanks.